Thank you again, Bjorn. I would now like to invite Alex to come back online, and he will, as Bjorn mentioned, present a more detailed view of the analysis tools in particular. So thanks, Alex.
Yes, hello. Let me share my screen first and go in presentation mode, hopefully. Nina, is it okay? Does it show?
Yes, it's perfect. Thanks.
Okay, so as Bjorn already introduced and mentioned, the analysis tools are tools that are not predicting B-cell epitope or binding or T-cell epitopes, but they're rather meant to assist in analysis of data sets, or either could be derived from the IDP or your own epitope set, and adding some additional dimension to these data sets. So here you see the menu option of the analysis tools, and the green flask means that some of these tools are in still a rather experimental phase, and we're still fixing them. So striking the balance between not releasing something that is too preliminary, but also making things available as we develop. So the population coverage tool is rather popular, frequently used, and what it does is, as stated there in the first bullet, calculates a fraction of individuals that are projected to bind or respond to a given set of epitopes. So as you know, for T-cell epitopes, one major factor is that T-cell epitopes are recognized in the context of HLA molecules or MHC molecules, which are themselves very polymorphic, and there are different loci and so forth. So, and these HLA frequencies vary in different ethnicities. So what you need to have is two data sets. One is one epitope or a set of epitopes with known characteristics. So you must know that epitope number 1 is presented by DR1 and DR5 and DR6, and or binds to these molecules. And then you must have your population of interest and know what fraction of those individuals express the HLA-DR1 and DR5 and DR7 gene and so forth. So to operate this, we rely on HLA genotypic frequencies that are compiled in different resources, in this particular case, allelefrequencies.net. So, for example, say I have a set of 11 different HLA MHC class II restricted epitopes. And I want to know what fraction of individuals would bind at least 1 or 2 or 3 of these different epitopes, and for example, in North African populations. And so I select here North Africa, class 2, and then I enter for each epitope which particular HLA molecules are restricted or binding, depending on the analysis I want to perform. And then the results come back and are either in a table format or in a graphic format. But you see here, for example, one measure is this PC90, which is basically how many which fraction of epitopes would be recognized by 90% or more of the individuals. the other of the individuals. So we have 11 epitopes, but of course, people have two different chromosomes. So the number of hits that are possible, it's up to 22. And so this is the percent coverage, what fraction of the individuals would bind at least one, and what is the average number of epitopes recognized in the population. So in this particular population, you will have 75% of the people would have would bind at least one of these, and on average, the average individual would bind 7. Another tool is a conservancy analysis tool. So this is a situation where you may have one or more epitopes, and you want to know, is this epitope conserved in a set of different proteins that you are want to analyze. So, for example, say you have identified one particular epitopes derived from the dengue virus, and you want to know how conserved is in a series of 20 different dengue virus isolates that I have obtained from a study in the field in Nicaragua or in Sri Lanka or whatever, or you have mine from the database. And so what fraction of conservation is also a function of what you called being conserved. So if you want to say how many this epitope is conserved in how many of this set of 20 sequences, you have to define, do you consider being conserved, you require 100% identity, or 70% identity is good enough, and so forth. So for example, in this case, we have a number of different, this is Lassa, so virus, a number of different epitopes, and then we ask, we upload a number of different sequences from the Lassa virus nucleoprotein, and then we ask how conserved this, and this, for example, in this particular case, we may say 100%. And so here you get this output of, in my 64 sequences that I entered, that I wanted to assess conservancy, what fraction of those 64 shows the epitope 100% conserved. And so these are the different epitopes, and they are conserved to a different degree here. And you see here the minimum and maximum level of identity. You can then, clicking on one particular case, have the breakdown of exactly what, where is the conservation and what kind of residues differences are found in the different isolates. And yet another type of tool is the cluster tools. So here is a situation, for example, where you have a set of experimental results from heterogeneous sources. Say that you have a series of results from different labs, and of course, different lab may synthesize different frame shifts of an epitope, or common also encountered in the case of eluded ligands, where you have essentially the same or similar related sequences, but they're not exactly in frame, or they may be slightly longer or shorter. And so you want a tool that cluster the epitopes in different groups Also, this is applicable in cases where you have multiple epitopes with different variants from different strains. We have used this type of tool quite a bit to generate what we call megapools or epitope pools, starting from large number of epitopes, boiling it down to a more manageable number of leads that are then synthesized and tested experimentally. So we have explored and implemented in the tool 3 different kinds of approaches. One approach is the connected peptides. And so here you say, well, I want to cluster peptides, and I want to have peptides clustered according to, say, a certain level of a threshold, for example, 70%. So that will cluster all peptides that are 70% overlapping, homologous to each other. Now, the drawback is that it will cluster things that not necessarily have the specified level of homology. And one clear example of that, say you are synthesizing overlapping sets of peptides. So 15 mers overlapping by 10, and that would have the 67% homology. So if you were clustered at 67% homology, a set of overlapping peptides, 15 mer overlapping by 10, spanning through a 200 amino acid protein, it will just return you a single giant cluster because they would be all interconnected, even though most of the peptides would not have 67% or 70% homology to the others. The other approach is this approach based on cliques, where you instead impose that all the members of the cluster, all the members of the club, must meet the fact that they have homology to each other higher than the given threshold. And this is nice, but the drawback of that is that one particular peptide can be found in multiple different cliques or clusters. And so to kind of find a solution to these drawbacks in the 2 methods, we developed a cluster break method where it is based on the first approach, but the cluster is broken down in subcluster based on a consensus sequence computation. And I will show you an example. So here are some fictionary sequences, trapper, clapper, snapper, wrapper time, daytime, anytime, per time, timeless, and so forth. Calamari. And the last one is actually the ovapeptide, the famous ovapeptide has no homology whatsoever. So what happens if we use the tool? So we enter these sequences, and then we specify what level of threshold sequence identity we want to specify, maximum, minimum length, and so forth. And then we go to the step 3 where we define the algorithm. So if you do all the connected peptides in a cluster, you get these results. So as I was saying, so with the first cluster that you get is this trapper time excess thing. And as you can see, trapper here has absolutely no connection with another member of the cluster, timeless. And that is the issue that I was mentioning earlier. So if instead you select the fully interconnected cluster, the clique method, you get this. So you get that all the peptides have a homology, but the timer, for example, can appear in two different clusters. And so the third method, the cluster break method, will give you this type of output, and where the clusters are separated based on the consensus sequence. And also here you see you have a graphic visualization of the methods where the different subclusters are shown, and you can vary this. And actually, if you mouse over, you will see which of the particular peptide sequences are represented by each symbols and what is the relationship to the other peptides. Yet another method analysis tool we have developed is this rate method, which infers likely restrictions based on HLA binding data and response data from a given population. So the tool is essentially conceptually similar to the methodology is commonly used to estimate some HLA association. For example, in autoimmune diseases, you know that HLA DQ 3.1 is associated with diabetes or whatever association. And what this means is that if you look at the frequency of, say, HLA-DR4 that is associated with rheumatoid arthritis, you look at the frequency of HLA-DR4, HLA-DR4 is more frequent in people that have arthritis as opposed to people that do not have arthritis. And this is expressed as, in a measure, is called odd ratios. So the same concept can be employed if you want to say what is the likely restriction element of one particular peptide. And so if you see that this peptide is frequently recognized by people that have expressed DR4, but is not frequently recognized in people that do not have DR4, you may infer this epitope as a possibility, but is restricted by DR4. So again, here you need to provide the allele data and the response data. So the allele data, this is your responding population. This is donor 1, 2, 3, 4, 5, 6, 7, whatever, with the HLA type of each donor. And then you will have the response rate. In this particular case, you provided ELISpot spot forming cell values, but could be categorical 1 and 0 or whatever other value, doesn't really matter. A big tool sees as response or no response. And in some cases, you might not have tested this particular peptide in donor 4 and 5, so that is labeled as not available. And then, without getting into a formula, the tool calculates the odd ratio and relative frequency value of the response in different categories. So you have the input data summary, and then you receive the restriction results in two different formats, which we'll discuss in a second. So the complete report is rather seldom used. It's basically for every single peptide HLA combination will give you the relative frequencies, and see in this particular case, for example, nothing is significant. Which, I mean, you might want to know, but there is no significant association. But the more frequently reporting format used is this one where results are filtered to give you only things that are have a significant p-value, even though this is not Bonferroni corrected. So if you want to have significant values with a Bonferroni correction, you would have to do it on your own. But so in this particular case, for example, you see you have this particular peptide in the context of DR beta 10101, and you see people that had that particular HLA and they responded to that peptide, there were 5 of them. And in conversely, 7 people did not have this allele, but also responded. So 5 out of 12 people that responded had DR beta 10701. While in contrast, the people that did not respond to this peptide, you see that you have only 2 people had the DR7, and the vast majority, 46 people, did not respond, did not have this number. So this is a significant p-value and noteworthy that not all the people that respond to this peptide are DR701. And in fact, the reason is because indeed this peptide is apparently restricted by both DR7 and DR1. And this dual promiscuous restriction do actually occur. Finally, the deimmunization tool. This is a tool that is often of interest of in pharmaceutical companies and biotech. The reason why is that often one makes antibodies or other protein drugs that are used as injectables, and in some cases, the recipient of the drug will develop an immune response against the drug, whether it's a monoclonal antibody or another protein drug, which is a problem, obviously, because that creates a potential safety problems or interferes with the drug's potency. So it is of interest to try to predict whether a protein drug has particularly strong epitopes, and also to predict which substitutions could be introduced to alleviate this problem and reduce potential immunogenicity. So this is what I was just mentioning. So the tools, it operates as follows. So first of all, you generate overlapping peptides from a protein sequence, and then predict the capacity of these different peptides to be recognized as dominant epitopes in human populations, and then suggest what particular substitution could be used to decrease binding. So in this particular case, we put in a sequence that we want to analyze. We define a percentile threshold, which can be varied. 20 is usually the default. And then here, the tool will give you a number of different sequences that are estimated to be potential promiscuous dominant epitopes. And so you select your epitope that you want to analyze. You also can select your email because it takes sometimes a while for the program to run and generate the results. And then the results are returned with a score. And let me elaborate a second on what is the score. So there are different outcomes. So first of all, you have whether the substitution has decreased the binding of the epitope per se. But one problem in introducing substitutions in the predicted peptide is you might inadvertently reduce the binding of the target peptide, but you might actually create an epitope upstream or downstream of that sequence. And this is what this score does. So apart from the reduction in predicted binding of the epitope, looks at the neighboring peptides. And so in the case of where you are just looking at one single 15-mers, obviously this is a not often encounter situations, but you don't need to worry about the fact that you will create a new epitope either at the N or C terminus of the peptide, because there is no other neighboring peptide. In the case where there is a sequence, but this is the best case. And then there are the other combination where either the one is absent and one is reduced, which is also good. And clearly the one you want to avoid is a situation where you created a reduced binding for your peptide, but you created 2 additional epitopes on either side. And so this is what the output of the algorithm is. So this is the reference wild type sequence, and then these are all the different substitutions showing you the percentile rank, and a decreased percentile rank is what you want, so a decreased binding. And this also gives you information of the predicted impact on the neighboring sequences. So to recap here, the different tools that are hosted in the analysis tools, the population coverage that projects what fraction of the individuals of a target population and are covered by a test set of epitopes with known HLA responses, the conservancy tool, the clustering tools, the rate tools that is used to infer HLA restriction, and the deimmunization tools that identifies particular substitutions to reduce binding affinity without altering in a negative fashion the neighboring sequences by creating additional epitopes. And with that, I think I'm returning.
Excellent. Yes, thank you very much, Alex. On screen. So we are running about 15 minutes ahead of schedule. Those presentations are in a little bit short, but that just means more time for the Q&A. So that's good, because I saw a couple more questions have come through. And Alex, if there's nothing else, I'll introduce Alba, if that's all good. Perfect.